Now that we've got some basic concepts down about gene expression, we can move into transcription of RNA from DNA. And so gene expression is the flow of information from DNA to RNA through the process of transcription, and from RNA to proteins through the process of translation. We're going to start with transcription and see how we can take that information in DNA and transcribe it into RNA. So first, looking at the differences between RNA versus DNA, there's some differences that I spoke about in previous PowerPoints, and I just want to remind you of those differences before we go forward. So in RNA, remember we have uracil instead of thymine. We have ribose as a sugar instead of deoxyribose, and RNA is single-stranded versus a double-stranded double helix of DNA. And so first, this uracil. So in RNA, we have adenines, uracils, guanine, and cytosine. And in DNA, we have adenine, thymine, guanine, and cytosine. And so there's this difference here between the uracil versus the thymine in DNA. Also, that difference in the hydroxyl group in the ribose sugar for RNA and the hydrogen that's on the number 2 carbon in the deoxyribose sugar in DNA. And also the single-stranded RNA and the double-strandedness of DNA. So the process of transcription involves some enzymes that are responsible for adding nucleotides onto that growing strand of RNA. And so now transcription is using one strand of DNA as a template strand to make a new strand of RNA. And the enzyme that's used to add nucleotides to that growing chain of messenger RNA, ribosomal RNA, or transferrin RNA is the enzyme called RNA polymerase. And so when we look at the DNA, and this DNA or the chromosome is separated into genes, we see that the area that is highlighted in red stands for that portion of the DNA that encodes RNA. So this red area is going to be our code for the messenger RNA that we're going to make. This RNA polymerase, along with some other proteins, are going to bind somewhere upstream of this gene. So right in front of the gene, the RNA polymerase will bind in this region, and then it will move along the DNA sequence and add nucleotides to make a new strand of messenger RNA. And so what happens is the DNA will separate, so those two strands will separate, and then the RNA polymerase will actually start adding nucleotides that are complementary to that strand, to one of those strands of DNA. And so you can see here in this region here that's been blown up, you see on the DNA, we have these nucleotides that are being used as a template. The RNA polymerase is going to add nucleotides that are complementary to that strand of DNA. So wherever you have a G on the DNA strand, you're going to have a C that's going to be base paired on the RNA strand. The same as if you have a C on the DNA, you'll see a G on the RNA. And wherever there's an A, instead of seeing a thymine base pair, you'll see a uracil, because uracils are the components or the bases of RNA instead of thymines. And so then the pattern will continue on until you get to a thymine here. You'll see that the base pair of the RNA will be adenine, because there are adenines in RNA. There are just no thymines. And so that's why some people get a little bit confused about what bases they'll be seeing in RNA. Remember, there are adenines, guanines, cytosines, and uracils. Just no thymines. So wherever you would see a T or a thymine, you replace that with a uracil. So the RNA polymerase is going to unwind a region of the DNA double helix. Whatever gene has to be transcribed is going to unwind the DNA in that region. And then the RNA polymerase will start adding nucleotides that are complementary to the DNA to assemble the RNA nucleotides on the DNA template to make that strand of messenger RNA, transfer RNA, or ribosomal RNA. The RNA transcript is then going to separate from the DNA. And so you see here, as the RNA polymerase unwinds the DNA, it's going in one direction, it's moving in one direction. And as that messenger RNA, transfer RNA, or ribosomal RNA is transcribed, those areas of DNA that have already been transcribed, they can recoil or rewind around themselves, and this transcription bubble will just move along the DNA until the RNA polymerase has completed transcription of that gene. And so once the transcript is completed, then it's released from the DNA, and the DNA is completely rewound, and then the RNA can travel to the ribosome in order to be translated. And so once we're finished with this process, we're either going to get mRNA, transfer RNA, or ribosomal RNA. And these three types of RNA are all transcribed in the same fashion. So messenger RNA, remember, messenger RNA is going to be that code for sequence of amino acids. It's going to tell us what amino acid is going to come next in the growing chain of protein. And it's going to give us the codon. The codon consists of 3 nucleotides. 1 codon equals 3 RNA nucleotides. The transfer RNA is the RNA that's going to deliver the amino acids to the ribosome during translation. The ribosomal RNA is part of the makeup of the ribosome. It's going to, the ribosome consists of proteins and ribosomal RNA. So in eukaryotic transcription, so transcription of eukaryotic genes, there is an initial primary transcript that's made. So if this is the DNA, a strand of DNA containing the gene that's going to be transcribed, it consists of exons and introns. And after that DNA is transcribed, all of the exons and introns are transcribed together. And so you see these areas where there's exons, introns, and then exons again. Once the messenger RNA has been has been transcribed, it must be processed, meaning the introns must be removed. So the introns are removed from this transcript, and then you're left with this final transcript where it just consists of the exons. And so this process of transcription is occurring in the nucleus because that's where the DNA is located. The DNA is in the nucleus, so of course transcription has to occur in the nucleus. But the ribosomes are in the cytoplasm. Remember, the ribosomes are closely associated with the rough endoplasmic reticulum in the cytoplasm. So the messenger RNA, after it's been processed and the introns have been spliced out, that messenger RNA needs to leave the nucleus through the nuclear pores and travel to the ribosomes so that it can be translated into proteins.